Much time is to introduce the speakers and let them speak. And then, as Mafalda suggested, we'll have a discussion at the end of the 3 talks. Okay, so any questions you have will be answered afterwards. So I think we have actually a very interesting set of speakers because they come from a variety of backgrounds and working also in different fields, all somehow related with this topic. So I'll start with Professor Anna Sebastian, who is a professor of the Faculty of Medicine, so close to us and close to the biomedical engineering, very close to the biomedical engineering students, if there are any here. They will for sure know her very well. She's the director of the Institute of Pharmacology and Neurosciences, I think that's the correct name, at the IMM here in Lisbon, and has been having lots of activities related with her fields, such as being the president of the Portuguese Society of Pharmacology, the president of the Portuguese Society of Neuroscience, and also a member of several committees related to these fields more internationally. She's been the director of the Brain Mind College of the University of Lisbon that has puts together several schools of the University of Lisbon, including technical as well. And she's been the coordinator of the biomedical engineering degree at the Faculty of Medicine. And she has very many projects that she is going to tell us a little bit about, in particular one of them, right? But which are addressing topics such as epilepsy and which drugs can be used to treat this. And also a very trendy topic, which is psilocybin and how psychedelics can also be used for therapeutical purposes. So I will let her talk about this. Maybe I'll already introduce the other speakers as well, because I think it's nice to see the variety of backgrounds. So next we have Dr. Filipe Barros, who's the youngest in the team. So we also have a variety of generations here, which is very nice. So Filipe is a postdoctoral researcher at the Champalimaud Foundation. She has a background in biochemistry, and she did her PhD in neuroscience in Paris. She won a prize for this very nice Prix de la Chancellerie de l'Université de Paris. And she is also currently a fellow of the Nel Mondi and Nessem Notchev fellow. It's a difficult name as well. As a graduate women in science, so congratulations for that. And she will tell us about the project she's working on at the Champalimaud Foundation on disease of motion disorders. I think it will be very interesting to hear about all the techniques from engineering that both of them are using in their research. And finally, we have Professor Paulo Aguiar, who's from my generation, so maybe somewhere in between. So, and actually also from the same degree. So Paulo graduated from physics engineering here in Técnico many years ago, like myself. Not so many, but yeah, I think they were born now. But, and he's currently, let me see if I say the right title, but yes, director of a group, the computational biology, sorry, I'm trying to look through. Yes, you tell us about neural engineering and computational neuroscience. Yeah. So as you see from the title, there is engineering, there's neuroscience, so very appropriate. And he works with a lot of very exciting state-of-the-art techniques to address neuroscience problems, also relevant to Anna and Filipe's work. So I think it would be very nice at the end to hear Paulo tell us about these technologies. Yes, he also has an incredible CV. I'm not going to go through all the details. You can read through it. And Paulo is in Porto at ISSS, a very big research institute, which has lots of different areas of research in health, but also neuroscience being one important one. And with this, I think I complete my introduction, and I leave us with the exciting part, which is the talk. So I ask, first of all, Anna, sorry to bring us her research. So I think you'll be using this computer, right? Yeah. And maybe you'd like to say here, right?
Oi. A sua apresentação é... Eu posso abrir?
Será que a gente fica do lado? Acho que deve ser essa. Acho que é essa. É essa mesmo. Ok. Okay, so Good morning, and thank you for being here, and of course, thank you for the invitation. And it is a pleasure to be here, and I always love to interact with more applied minds and engineering, being bioengineering, medical engineering, whatever it is. But so it's always a pleasure to be at IST to talk with students and to share our interests. So I'm going to speak about a recent project that is nearly finished that we have. And the reason I chose this project is because we use different techniques and approach that may be useful for you to understand what you do a little bit in the lab. There are other projects that I keep aside, as Patricia mentioned, so we are also interested in understanding what psilocybin does in the brain. This involves lots of behavioral actions and proteomics and transcriptomics. I'm not going to speak about that. I'm going to speak about a project that we have that aims to understand the mechanism of action of a specific drug that was supposed to be, and is supposed to be a potential anti-seizure drug. Of course, just still in animal research. Besides these projects that are more connected with me, the group is a large group, so I didn't brought the slide. But anyway, we are also interested. There is a group with interest on neurogenesis that has some collaborations also with Technico, with Margarida Diogo, yes, Professor Margarida Diogo. So neurogenesis, postnatal neurogenesis and neuronal development. There is a group very much involved with neuroprotection and neurotrophic factors, and there is a group very much involved in neuroinflammation, and another one, of course, small groups in the big group, very much interested on the crosstalk between astrocytes and neurons. So there are several projects in the unit, but we have to choose, and I choose this one to show you. Okay, pass the slides here. Okay, so the very beginning of it was a paper that appeared by a colleague at NIH that I know very well, Ken Jacobson, that is from medicinal chemistry, and so designs lots of drugs. And he identified, so he has been in the adenosine field, is a field that I like, is a neuromodulator that controls many activities of the brain. And he designed this drug that was supposed to be an agonist, a ligand of a specific receptor for adenosine, for this neuromodulator, with anticonvulsant activity. Okay, so this was not really challenging, but really what puzzled us is that this drug has an anticonvulsant activity in mice, of course, in animal models, but did not have cardiac side effects. And I will explain why this was puzzling. This was puzzling because, well, adenosine is a neuromodulator, is an endogenous substance. By the way, when we take caffeine, you just had coffee. When we take caffeine, what we do is to prevent the inhibitory actions of adenosine in our brain or to reduce them. So that's why we wake up and we have a little bit more aware of things. So adenosine has really an inhibitory action in the brain, and because of that, it is considered also an endogenous anti-epileptic agent. So drugs that would favor this activation of the A1, the inhibitory receptor, are expected to have anti-epileptic action. So no novelty in that, but really the strange thing was, and that a drawback with and what has been preventing further development of drugs related to adenosine as anti-epileptic or anti-seizure medication is because of the cardiac side effects, because exactly the same receptors that are in the brain to control and to reduce neuronal activity, we have receptors in our heart that also control heart rate, the force of contraction. So usually in vivo, substances that activate this receptor also decrease heart rate, also decrease the force of contraction. So these side effects of adenosine-related medications are, okay, I should ignore what is here. I think so. What is here? Okay. Okay, let's close it. Where should I close it? Okay, it's again back, I hope. Okay. So we really, what we asked and what I proposed to Ken Jacobsen was to start a project to understand the mechanisms of the anti-seizure action of this specific drug, and what makes it different from other drugs that activate the same receptors for adenosine. Okay, so we started by doing very, very simple experiments, and so to prepare hippocampal slices and to test what this drug would do. So the hippocampus is in a brain area of a mice that is quite easy to isolate. We cut the hippocampus in small pieces, in slices. We used electrodes, and in this particular project, what we started to do was to do patch clamping in pyramidal neurons. That is to say, to understand how the drug would affect communication between excitatory neurons and this neuron from which were recording. Okay, where is it? Okay, this. Okay, I have to see here. Okay, this neuron here. Okay, so, and really the big surprise, so this was first big surprise, is that this drug does not affect excitatory synaptic transmission. And this is really in clear contrast with what happened. This was a master thesis of a student, and I was not even believing this, and asked her, well, please use a classical A1 receptor agonist to see if there is a problem, technical problem, or if it is a drug problem. And this is very sensitive. And really, so the classical, let's say, A1 receptor agonist cause a clear-cut inhibition of synaptic transmission. So synaptic currents before and after the drug is quite clear that there is an inhibition. But in this particular drug, there was no inhibition at all. So I was so surprised that we even asked. So probably this is because we are isolating very much the system, because to record synaptic currents from a neuron, just from a neuron, we need excitatory ones. We block the inhibitory currents. In a way, we isolate very much the system. We force the membrane potential to be in a specific level. So what if we could record just in a more integrated way and free system so that we could record synaptic potentials without forcing the membrane potential into a specific level, without blocking the biologic transmission, and in a more, let's say, physiological state. So again, in hippocampal slices, exactly the same, but instead of recording just from one cell, we recorded from a population of cells that was nearby the electrode with field recording. So we are not inside the cell, it's just a field recording. And again, there was no effect of the drug. And we tested several concentrations up to 0.5 micromole. It was already a huge concentration. No effect at all. And again, an effect, of course, of what we always see of a classical receptor agonist. Okay, so this was the first conclusion, and we are really puzzled about that. This drug, though being considered a typical, so an A1 receptor agonist is not a typical one, because it does not affect what every A1 receptor agonist does, as far as we know. So we thought, well, this drug has anti-seizure action in vivo. This was proven in that paper. But so can we, if we use, let's say, an epilepsy model, can we see any effect? And the answer was yes. So we started by using slices again, but slices in culture. So the slices, when we culture slices from animals, newborn animals, and we put them in culture, and just doing small tricks with the medium that is in culture, after 2 weeks, more or less, they can be used up to 3 weeks, but after 2 weeks, they start to develop this spontaneous activity that can be recorded. And this is a kind of ex vivo model of epilepsy, because there are these bursts of activity, spontaneous activity, so we're not doing anything to the slice, just recording from it. And we tested the effect of the drug, and yes, there is an effect. So the frequency of the events within these bursts is decreased by the drug, okay, in the experiments we did with the slices, organotypic slices, but well, when we did not, this is the control of time, because of course time matters. If you didn't apply the drug, the situation, so the slices were quite stable, but with the drug, before application and after application, there is a clear cut inhibition. So in an ex vivo model of epilepsy, the drug works. And we then asked, well, and if you use epileptic slices, so it's not organotypic slices, but we made the rats epileptic before, and then we isolate, of course, we killed the animal 1 month after inducing epilepsy, cut the slices, and recorded the field recordings that I showed you in the second slice, from a slice from an epileptic, let's say, animal, and really in these animals there is an inhibition of these field recordings, again in clear contrast with the control animals where there was no effect at all. Okay, so the drug works, but works only in epileptic-like situations. But still we didn't have a clue what was the mechanism yet. So then we start asking, well, probably there is no effect on excitatory synaptic transmission, and we can see an effect on epileptic animals when we have the system more free, so we don't block anything. So probably what we have is an effect on inhibitory synaptic transmission, not on excitatory synaptic transmission. And so this is what we tested. And so going back again to patch clamp, but we recorded from inhibitory neurons, okay, and stimulated other inhibitory neurons. So we were recording inhibitory inputs over inhibitory neurons. And this was, so we tried several combinations, inhibitory inputs over excitatory neurons and all the combinations, but this was the one where there was a clear effect. And so that the drug was inhibiting, so before and after the drug, the currents, the inhibitory currents that we could record, the drug was inhibiting the inhibitory inputs to inhibitory neurons. So if you think that if we are inhibiting the inhibitory inputs to an inhibitory neuron, we are disinhibiting this neuron. This neuron will work. This neuron would work more, so is inhibiting the excitatory neuron more efficiently. So this may explain. So a selective action of the inhibition of the inhibition may explain why the drug was in a way disinhibiting the inhibitory neurons that project on the excitatory ones, and could explain its action on epilepsy, its anti-seizure action. So this was one mechanism. This is one possible mechanism that still holds. Another one that is related to the inhibitory neurotransmitter, and I didn't mention it yet. So the main inhibitory neurotransmitter in the brain is an amino acid that is called gamma-aminobutyric acid. That's called GABA. This is the main inhibitory neurotransmitter in the brain. Actually, it is the neurotransmitter that inhibitory neurons release when they are inhibiting other inhibitory neurons or when they are inhibiting other excitatory ones. So, and we know that this neurotransmitter is released to the synaptic cleft, and we know that there are molecules that take up the neurotransmitter that in a way end the time of action of the neurotransmitter in the brain. And these molecules that are called GABA transporters are very important and are targets for anti-seizure drugs, because if we block them, more time this inhibitory transmitter stays longer in the synapse, and it will of course promote more inhibition. So, and the question we asked was whether this drug could affect GABA transport. So to study GABA transport, we use a neurochemical assay, a neurochemical approach, looking at the uptake of the neurotransmitter tritiated, so radioactive neurotransmitter, and then, but in slices, we incubated and we washed it out, and we could read the amount of radioactivity kept in the tissue. And really the answer was that yes, again. So this drug can clearly inhibit, these are traces in different experiments in the absence and in the presence of the drug, there is a clear inhibition of the ability of the transporters to take up GABA, so to end the action of GABA. Of course, the averages, so the effect is significant. And another surprise was that the effect of this drug, what we have here is percentage change of the ability of those molecules to take up GABA. And so the effect of the drug that I showed you before, this is absolute values, this is percentage inhibition as compared with the control, the drug that we are testing. Okay, the first big surprise is that the effect of this drug was not affected by a classical A1 receptor antagonist. So when we combine the pharmacological combination, the drug testing that with the A1 receptor antagonist, the effect stayed, so it was not blocked, but it was clearly blocked by an antagonist of another receptor that is also an inhibitory receptor, is not very much expressed in the brain, but it is there. So it is clear that the effect of this drug that we're testing was blocked by the antagonist of that receptor. And the effect was mimicked by a substance that activates that receptor. So we could conclude that the effect of this drug is mediated by the ACE3 receptor, so is the other inhibitory receptor, but not the A1. So the last question we asked, so why the effect was mostly evident in epilepsy-like conditions? We knew that these receptors are not very much expressed in the brain. So the question we asked, well, are these receptors in a way overexpressed in a disease situation? Because it is quite common that during diseases, there is change in the expression of different molecules in the brain. Some of them are really cause more harm. Others are kind of attempt of the brain to control the situation. And so we asked whether it could be an overexpression of this receptor in the brain. And we don't have yet the data for the rats, but we started with the human tissue because we have human samples, tissue samples in the lab so that they could be tested right away. And really, so this human studies are very difficult because the controls are very difficult. So the controls here was post-mortem brain tissue collected. It was in the Amsterdam Biobank, so they can collect the post-mortem tissue quite few hours after death, in clear contrast with the southern countries where the tissue can only be collected 24 hours, so it causes lots of problems. But anyway, these are the controls, and this is tissue from epileptic brain, patients with epilepsy, but from surgery. So with all the cautions that is comparing a post-mortem brain with the brain taken during surgery, anyway, there is an overexpression of this receptor that we identified with classical Western blot techniques with an antibody. So we are now doing it with rat tissue that is much more controlled, so we can have the exact same animals with and without epilepsy. So, but this data I don't have yet. But yes, so it seems that there is an overexpression of these receptors in epilepsy, and this is quite interesting because really this may highlight that these receptors may be nice targets for anti-seizure drugs because they are not very much expressed in healthy brain, and we would like to think that also not in healthy neurons. And probably if they are overexpressed in neurons that are firing at high frequency, it could target those neurons that need to be targeted without having other side effects, such side effects as most of the other anti-seizure medications. Okay, so this was it of this project. Of course, Ken Jacobsen from NIH and Tosh, so those that synthesize these nice drugs, and people that worked in the project during several stages The initial electrophysiological data was from Diogo and Katia Domingos. And the remaining experiments was from with Anuisha, the uptake studies with the help of Tatiana, and saw the inhibition of inhibition. So the studies on patch clump studies on inhibitory neurons. Okay, and of course financing, that is so important. All engineers know that. So thank you for listening, and that's it.
Thank you, Anna. I think we decided to have questions at the end, right? So I'll ask Filipe now to join. Thank you for this detailed dive into the drug. That was really interesting.
So, welcome everyone, and thank you so much for inviting me to speak here today. Throughout my presentation, I'm mostly going to talk about my professional journey and path and some of the experiences that I've had so far. So I'm not going to focus on one specific project. I'm just going to share with you because I want to share my experience so far. And I want to start by talking about my very beginning, which was in 2011 in Porto. I'm from Porto, and that's where I started studying biochemistry. So I'm not a bioengineer. I'm a biochemist by trade, as Professor Patricia already mentioned. And then I had my first international experience in Sweden at Uppsala University. So you may have heard of it. It's compared to Coimbra here because it's really a universitary city. And then I came back to Porto to do my master's again in biochemistry. And in our last year there, we are supposed to do an internship, which is from 9 to 12 months long in order to carry out our master thesis. So I chose to go to Paris and did my thesis there. And I fortunately got invited to stay and continue my work and do a PhD there, which I finished in 2020 and then came back to Portugal, now to Lisbon. I'm now an Alpha Senior recently. My sotaque is still present, so yeah, I'm not going to hide it, and I'm proud of it. Since 2021. So just to show a few pictures of Uppsala University, it could be like this, but it also could be like this. So very snowy. I went in January and then came back in June, July. So I really got a period from when you don't have any sun, which more or less it's like 3 hours of sunlight per day, to the period where it's much more sunny like this. You can see the banks with a lot of snow and then no snow at all, which is always sunny. And then you wake up like at 4 a.m. in the morning because that's when the sun starts to rise. Yeah. So I got both ends, and this is the first lab that I was in at the molecular and cellular cognition group, because even though I was studying biochemistry, I was really passionate about the brain, and this is what I wanted to study, and I had a real clear vision that this is what I wanted to pursue. But at the time, in Porto and like near Porto, and I think mainly over Portugal, there was no program in neuroscience. So from bachelor's to master's in neuroscience. So I figured to have a generalistic scientific background, which biochemistry did and served really well, and then go and specialize in neuroscience. So for my first internship in my bachelor thesis, that's what I did. And I studied the characterization of the zebrafish COMT homologue. COMT, or catechol O methyltransferase, it's an enzyme that degrades catecholamines. Dopamine is one of it, and it's involved in many functions in the brain, namely psychiatric disorders when there's an imbalance of the levels, but also in movement disorders. That's what I'm studying now. And I was working with zebrafish, so that's my first animal model that I started with. And then, as I mentioned, I went to Paris for my master's internship. And I mainly chose this specific lab, and I was really focused on it because they were studying vestibular science. So inside our brain, we not only have the brain as like most of you think about when you think about the brain, but we also have very important parts that are the inner ear. You have the cochlea and the semicircular canals, and the semicircular canals, it's what keep you balanced, because we have a fluid inside, and then there are 3 of them, and then detect the angular acceleration as the fluid goes throughout the canal. And due to personal experiences, one of my uncles had a vestibular disorder, Meniere's disease. I was really fascinated about it, and I wanted to know more about this. So I chose the Spatial Orientation Lab in Paris. And you can see here the picture of the building. But I also wanted to share with you where I lived, because I think that's also important, right? We are always focused on our studies and what we want to achieve in our professional career, but we also must enjoy our way until we get our goals right? Because it's also about the journey and the ride. It's not only about reaching the specific goal. And I was very lucky to have a place at the Cité Internationale Universitaire de Paris, or CIUP for short, which is home of over 12,000 students of over 150 nationalities. And I lived at this beautiful house called Maison du Portugal. And most of us were Portuguese, so it was my home away from home, because I was in Paris for 5 years. So as you can imagine, you get homesick a lot, but I had great colleagues there, Portuguese, but also of around the world, and to share these 5 years with, and it's amazing. And if you ever consider going and doing studies in Paris, you should definitely consider living at CU. So then, as I was mentioning, I wanted to study vestibular science, and to do so, one of the main things and one of the main outputs that you can study regarding balance is gaze. Okay, so gaze stabilization was the focus of both my master and PhD thesis, specifically in mice. So I went from zebrafish to mice. And then the adaptation of visual vestibular inputs and predictive motor signaling during locomotion. So this can sound a bit tricky, but I'm going to tear it down in pieces and you will get it. So as I was mentioning, gaze stabilization is one of the main inputs we can record, because as you can imagine, the inner ear is not really very accessible. But we have 2 main reflexes that are complementary, which are the vestibulo-ocular reflex, or the VOR for short, and the optokinetic reflex, or OKR for short. And the VOR reflex responds mainly to the vestibular signals, as I was saying. So when you turn your head quickly, for example, to the right, your eyes will turn exactly in the same magnitude, but opposite directions to the left, okay? And this is it's an involuntary response, okay? It's a reflex. And the optokinetic reflex, or OKR, that responds mainly to visual inputs. So I'm sure you have all seen this. I've ever been in a train or in the metro or whatever, and you're sitting, and then if you look and your metro is still, but the next metro, like on the other track, is moving, your eyes will go like this, but you are still, right? So it's this is the optokinetic reflex, okay? So now you know how it's called. Next time you see it, you can share this piece of science with your friends. And it's composed of short and slow phases to reposition the eye in the orbit. And we do this to maintain our balance, both the VOR and the OKR. As you can see, there's a slow phase and then a quick phase that is done to reposition the eye in the center of the orbit. So how do we test this in mice, right? We did so using the video oculography technique, and I'm going to share with you until the rest of the presentation some of the bioengineering, I would say, techniques that I've used and I'm using. And the video oculography is one of them. So just as a curiosity, in the studies that I did, we were using this kind of futuristic helmets in mice. We implanted these helmets with stripes, right? Because you can imagine that the stripes are creating a visual vestibular mismatch, right? Because if you have in front of you, have you ever put on like a friend's glasses just for the joke? And you're like, whoa, because you cannot, like, if you don't see correctly and there's something in front of your eyesight, you cannot stabilize gaze properly. You need to adapt. So there will be changes in your reflexes. And this is exactly what we're doing with this helmet. We're putting stripes. It could be other things, but stripes are in the literature are already known to have or to cause an adaptation in these reflexes. So this is what we did. And we fixated these helmet-like devices in mice for 2 weeks. So they lived, they ate, they drank with it. So they lived normally with this helmet. And then we take it off and we tested the reflexes. So both the VOR and the OKR after they have worn this. And we use so doing, to point, maybe it's not that great, but a vestibular turntable. So it's like an enormous apparatus and a table that is turning, and the mouse is sitting right here on this tube, as you can see this representation. And we have cameras that are that can acquire the eye movement in with a lot of frames per second. I'm gonna just play the video again. And as you can see here, this is an example of the VOR. So the table is turning and we're recording the movements of the eyes. And this is exactly what we have. So this is the head position because the table is going like this. So there's an undulation of the movement, and then the eye position in response to this undulation. And this was for the OKR. So as I was mentioning, for the OKR, we are still, and then we have some visual motion that goes around our environment. And we simulated this with the mouse was still in the same table, but it was not moving this time. And we put on a dome and we projected with a projector, like tiny dots that you can interpret them as like stars. For example, these things like babies have for their cribs, where they project stars and the space in the ceiling. So sort of like this. So the animal follows these dots, and we could also record the OKR. And then for the predictive motor signaling during locomotion, I still used video oculography and among other techniques, but I chose this one to share with you to study the reverse. So what do I mean by this? Usually we think that it's always the brain that commands the inputs that go to our spinal cord, but that's not the case. And the spinal cord has a very important function and quite primitive, and it's conserved along a lot of animals throughout the animal kingdom. In this research, we did studies from the xenopus, so the frog, until the mouse, and they both had this new pathway that we unraveled, which is a spino-ocular pathway. So from the cervical spinal cord up until the vestibular nucleus, that is commanding eye movements even when the animal is decerebrated. Okay, so the animal here in this video where you're going to see has no brain. Okay, it only has the brainstem and the spinal cord, and it's still moving its eyes. So you see how important the brain at the end of the day is, which is very important, but it's mainly its function is mainly to modulate a lot of intrinsic and primitive signals that already exist in the spinal cord. So maybe it's not working, I don't know. But here you will see a mouse running. Okay, it's fine. So these are the eye movements like I showed you in the previous slide, and this is a mouse on a treadmill, and we were recording with the video oculography technique the eye movements while the animal was running. And may I remind you, this animal has no brain, only brainstem and spinal cord. And you could see, if you notice, these eye movements that you just saw on the other side of quick phase to reposition the eye in orbit and the slow phase on one of the directions. And you see the animal is running, there's these eye movements, and then it's slowly gonna stop running, and the eye movements start decreasing. And on the right is just a panel of the paper that we published describing this spino-extraocular pathway, where we have, on top of the eye signals, we also have the locomotion that is tracked using neural networks that I'm going to show you afterwards. And the eye movements described and the phase that between movements of the eyes and the movements of the paw. So another very interesting experience I had during my time in Paris was being able to go to a parabolic flight, because my lab, my team, did experiments in weightlessness, so microgravity. And I brought this video to show you how does it feel. I took this video from the window of the plane, and you start to see that the wings, this is not a normal position, right? So the plane is already doing a parabola, right? You see the skyline, how it is in the angle in relation to the plane. And then they shut down the motor, and then you start doing the parabola down. And this picture right there on the right, it's one of my lab mates, Michele, that was the one that was doing these experiments in human subjects, so this was not in mice. I was one of the subjects of the experiment. Yeah, I was one of the mice. And we had this, the goal of the experiments was to test haptic perception in weightlessness. So we had to reproduce a cube that we are seeing in this like Oculus, Google Oculus. I don't know if you have heard about it. And we had this lever, and we had to reproduce it. And it was very interesting because we were alongside real astronauts. For example, Thomas Pesquet was driving the airplane. Also, Matthias, one of the German astronauts, was training for his period in the ISS. He was right in front of us, and he was training how to plant plants in a vertical garden, because they want to do that in space. So he was trying. And each flight that we had, we were there over 3 days. So we had a flight per day. We had 90 parabolas, and the parabolas lasted like around less than a minute. So you had to really take advantage of this time to start your experiment and with your subjects ready and everything. But it was definitely an amazing experience, and yeah, that I will for sure keep always with me. And right now, I'm a postdoctoral researcher at Champalimaud Foundation since 2021, and I'm studying a movement disorder called dystonia. So dystonia is characterized by involuntary intermittent muscle contractions. So you can imagine, for example, you have this arm and both both muscles are contracting. The antagonist pairs of muscles are contracting. So you cannot control the types of movements that you're doing. For example, you have here the feet of a patient, and then this is called focal dystonia because it only affects one limb. And then you have a generalized dystonia where you can see that the back and also the neck and the arms are affected. And we're doing so using a mouse model, a genetic mouse model called DYT-TOR1A, because TOR1A is the protein that is mutated in this genetic model. And we are using in vivo techniques to see how the neurons behave in vivo. So here, I don't know if the lighting is all right for you to see, but we have a coronal view or a coronal brain slice. And here is the striatum, which is the area of the brain that we are studying. And we put lenses that I will show you right on the next slide in order to see the neurons lighting up while the animal performs a behavioral task. For example, here we have depicted the reach to grab task. So the animal has to reach out for a pellet, because as you imagine, we want to reproduce scenarios where the muscles are activated, and we would want to regulate, excite or inhibit these muscles. So this is a technique that I was mentioning with the lens. So this is called in vivo calcium imaging, and we use miniature microscopes, really, really small. The proportion here is correct, even though this is a drawing. The size of the microscope is correct in regards to the animal. And we implant a lens in the striatum, and also by injecting previously adeno-associated viruses with fluorescent proteins that are only going to bind, because they have the Krylov system, to the neurons that we want to target. Specifically, we want to target dopaminergic, we want to target D1 and D2 neurons, so different populations, because we believe there is an alteration in these pathways, and they will only connect to this D1 or D2, these viral vectors with the fluorescent proteins. So then when the animal moves, we will see flashes of light, fluorescence, of these neurons that were infected, if they are active, right? Because I think you know that calcium passes through the synapse when the synapse is happening, or the communication between neurons is happening. So it's a proxy of neural activity. It's different, of course, from the electrophysiology recordings, which are the real ones, let's say, that we can see potentials and everything. Here we are only seeing fluorescence differences from the baseline, but it already tells us if the neuron is active or silenced. And then we also use things to measure movement, which some of you might already guess, accelerometer, because we want to know how fast and how many Gs, etc., the animal is moving. And also electromyography, which is used to record muscle activity. Here you have a small PCB board and with the electrodes that will be inserted in the muscles that we want to study. And also we join this with deep neural networks for assessment of the dystonic movements. So in here you are seeing a test that it's called the open field test, where we put the animal in this open field and we just let it behave and do its thing for a certain amount of time. And we're studying naturalistic movements, right? We are not forcing them like in the other tasks to reach for something. Here it's a different paradigm. We are just letting them be and see what happens. And we use a markerless post-decimation technique that is based on deep neural networks called deep lab cut. Some of you may have heard about it. And these small colored dots that you're seeing, it's markers that are placed offline where we train the neural network in the areas of interest, and then we're able to track where the animal moved. So if we combine everything, we have something that looks like this, which is the input from the accelerometer. We have body acceleration, whole body acceleration, and also the angular velocity. The open field test while the animal is doing an open field test. And also we have the neural activity that's on the right. And when you see flashes of light, it's because the striatum was lighting up. And underneath there's the regions of interest. I don't think you can see it very clearly because of the lighting, but each of the colors represents a circle that's made around one of the neurons, and every time there was a movement here, it means that the neuron is flashing and it's activating. So this concludes my presentation. I wanted to share with you that we are hiring. So if you're interested in joining our lab, please come and see me. We have open positions for master students that want to do their thesis with us. So feel free to talk to me at the end of this session. And I've placed also my contact, both my email and Twitter and a lab page for you to look at in more detail. And I just want to mention that my group is very nice. We're a very nice bunch of people to work with. It's a mixed group, so we have 2 clinicians, master students, PhDs, research assistants. So it's really a mix of backgrounds that is working really well, and we are a recent team and we're eager to go for it. So please join us. And thank you so much for the opportunity.
Thank you so much, Felipe, for an overview of your track. And now Paulo, for last but not least.
Okay, thank you, Mafalda. Okay, so good afternoon, everyone. It's really a pleasure to be here. Thanks so much for the invitation. And it's also nice to be back at IST, because this was my home for some time ago when I was doing my first degree. So I've been asked to talk a little bit of what my group does, and also address one particular project that I could fit in like 15, 20 minutes. So I'll try to address both things. And to put you into the context of what is the pillar of our research in my group, let me start with a question. So why do you think we have neurons? Okay, what are neurons good for? If I would need to collapse the definition of nervous system in one word, which word would you choose? And it's a question for you. So if I would ask for the skeletal system, maybe I would say structure. If I would ask for the muscular system, I would say movement. Which word would you choose for the nervous system? You can say louder. Communication. I like this one. Another option. Sorry? Okay, more stuff. No? I like information, okay? So I will try to convince you in a couple of slides that the nervous system, and neurons in particular, are nature's solution to the problem of acquiring, transmitting, storing, and processing information. And this is in the key of what we do in our lab, okay? So we try to understand information processing in neural circuits. And we do this using what we call bottom-up neuroscience. If you talk with a neuroscientist or someone working in neuroscience, one of the first things that you have to ask is, at which scale do you work? Because one of the nice things about neuroscience is that you can work at scales of nanometers in the synapses, up to meters on axons on your spinal cord, in milliseconds, up to years. So there's a bunch of scales. We work on bottom-up neuroscience, where we like to assemble small populations of neurons and try to understand neural computation. Okay, so neural computation. We work both in fundamental neuroscience, where we try to contribute with new knowledge on neurobiology. But the thing that brings me today here, I suppose, is the development and use of different methodologies to reveal and repair neural function. So neuroscience is no longer the backyard for neurobiologists. Neuroscience needs informatics, engineering, computer science, mathematical modeling, and all of this to really face today's challenges. Okay, so most of the problems nowadays cannot be solved just with molecular biology and more traditional methods. If I'm really interested in neural function, unavoidably, and as you saw with Ana Sebastião and with Filipa, we need to address electrophysiology, okay? So the signals that neurons will be sending to each other are in today's paradigm, on our understanding of neuroscience, most information is being carried out in terms of electrical signaling. So for us, it's very important to look at them, monitor them, modulate them. And I will talk a little bit about the techniques that we use. One thing that it's interesting is that if we are using with these small neural circuits in our lab inside dishes in vitro neuroscience, we have the type of signals that you have on the right, but our lab also deals a lot with the signals that you have on the right. And the difference between these two signals is that these are extracellular signals on in vitro populations or in organotypic populations, and the ones on the right are from electrodes that are implanted in patients from either epilepsy, or we also work with electrophysiology from patients that have Parkinson's disease, so that they have deep brain stimulation. So we have very interesting ongoing collaboration with clinicians, helping them out and exploring this type of data. But for today, I'm going to address just the left ones, okay? I will be talking about microelectrode arrays. Microelectrode arrays can be used in vitro and in vivo. In here, I will just address in vitro. And basically, you can see them as an array, so a grid of tiny electrodes. They can be as small as 8 micrometers. We usually use with 30 micrometers. And they cover a chamber where we can put our neurons on top of them. It's not as precise as patch clamp that you heard Ana Sebastião talking about, because with patch clamp, we can know everything about the single neuron. We can go to subthreshold potentials and acquire all the information about neurons individually and ion channels and so forth, so on. But the precision that you have in patch clamp also plays against if you want to know things at the network level. If you want to measure several points at the same time, this is the way to go, okay? And our system has something like 6 electrodes. We can both record and stimulate on all of them. So it's a good way for you as an engineer. Once we put our culture in top of the mayor, we put on our hardware, we sit down in the chair, and we are engineers from now on, okay? So we program the electrodes, we tell the electrodes how to stimulate, we record to modulate the network. We also do this in a combination with microfluidics, because as I told you before, we are very interested in bottom-up neuroscience, a little bit like our colleagues on engineering that they assemble, let me put a transistor here, let me put a capacitor, an IC, and let's try to build function. We also try to do this but with neurons. So we need to compartmentalize and say we have neurons sending this information, we have inhibitory neurons here, and so forth, so on, to be able to generate function. I'm sure you've heard about microfluidics. We combine them with the microelectrodes in what I call the micro-EFs. So this thing got the format's not well, but anyway, we combine these microelectrodes with the microelectrode arrays, and what we can do is either cultures that only have neurons, but we can also combine neurons with other cell types and really force circuits. In this case, for example, we make very tiny microchannels where only axons can go through. Neurons only exist in one side. We force the axons to go through those microchannels that have electrodes underneath, and we make these axons synapse either to other neurons or other cell types. And we can control everything that is going on the axon. So we can listen and modulate those signals. We can do this in combination with calcium imaging that you heard Filipa mentioning about. Her stuff is much more difficult because they do it in vivo. We have this under a big microscope, not the tiny one, but still we can combine the stimulations of the electrodes. You don't see them properly, but you see a black circle in here and maybe another one in here. We can trigger activations on the axons when we want. And we have this amazing amount of detailed data to inspect what's going on. It's really a pity that it got maybe it was when it was copied to here, got the format. Anyway, still we can read it well. This slide is here for a message that you guys as bioengineers, I think it's relevant for you to know. This can generate a huge amount of data. And if you want to innovate in science, most of the time you end up in two situations. Either you have access to instruments that no one else has, and you can see data that no one else has seen before, okay? That's very expensive. You need to have money to get those really expensive equipment. Or you guys can see things in the data that no one else sees, okay? And machine learning is not the answer for everything, okay? It's in the hype. We have publishing stuff in machine learning, but use your human learning is also good, okay? So try to use your natural intelligence instead of just the artificial one. We also contribute a lot with these computational tools. We really like this, this challenge of analyzing this large amount of data and extracting information. So one of them is this microspike hunter, which looks at all the signals that are being transferred by the axons and try to do decoding, for example. So how do neurons communicate with each other? It's not like Morse code, right? It's not like Morse code, but they do send the spikes. How can we know which information is being sent depending on the train of spikes? So we can analyze this kind of things. The small, tiny example of project that I want to tell you about today is, so this was just an overview of things that we do at the in vitro scale. Now, a very specific project that I think would be of interest for people in bioengineering. So closed loop control and what the community calls hybrid systems. So if I convinced you that neurons are good for encoding, transmitting, storing, and processing information, I think you will also agree with me that most of what's going on in terms of your behavior has a counterpart in terms of spatial temporal patterns of activity. Okay, so if you're looking at those neurons and the specific pattern is activated, maybe you're with your arm like this. A different type of pattern will mean that you are doing something else. So you have so the patterns of activity on your brain are extremely relevant. And they will be distinct depending if you're in physiological or pathological states, okay? So, for example, Parkinson's disease will have very specific, what we can call electrophysiology biomarkers, which can allow us to understand that this circuit is not working properly, so there's something wrong in here. If diseases and physiological and pathological conditions are so tightly connected to these patterns of activity, why don't we try to modulate directly the patterns of activity in a way to achieve therapeutic strategies, okay? So in an alternative to pharmacology, why don't we modulate neuronal activity? This is not new. I mean, people have been doing this for tens and tens of years. You may know about cochlear implants and the DBS, but this is, in fact, a very interesting strategy to address some pathologies. Now, the current technologies for neuromodulations are still far from perfect. To start with, there's an idiosyncrasy of the battery. So you guys may need to go to surgery just because of replacing the battery. So that's a big idiosyncrasy. Also, most of the devices now, they work on open loop control. You're in a school of engineering, for sure you know what's open loop and closed loop. So most of the ones today work on open loop. And they are clock-driven computation. Clock-driven computations like our CPUs. They have 3 gigahertz and everything needs to be done on that clock. The alternative is event-driven computation. This is how your brain works. So it would be nice to have our medical devices also working on event-driven computation. Like I mentioned, they also have this constraint of using lots of energy. Battery is a big concern, and usually they are bulky. Bulky to the point that in DBS, you have the electrode implanted here, and you have the wire that comes down, and you have your APC, the stimulator on your chest. So we are trying to figure out new ways of new devices to try to monitor and modulate neural activity based on new types of devices. And I'm going to tell you about the memristor. Okay, the memristor is a very recent device. When I say recent, is 2008, something like this. It has been hypothesized for already some years, but the IBM just made it very recently. You have two definitions for memristor, but the one that I'm going to use is the two-terminal non-volatile memory device based on the resistance switching effect. I'm going to deconvolute this thing now, but basically this is a neuromorphic device. I'm not sure if you have heard about this concept before. Neuromorphic devices are electronic components that have dynamics that are similar to specific structures of the brain. And what's so special about memristors? Let's remember your IV curves from Ohm's law. Okay? In an IV curve from Ohm's law, you have your current is proportional to your voltage. Okay? So in a memristor, instead of having a line, you have an hysteresis curve, which means that the conductance depends on the history of what has been impinging on the memristor. Guys, it means that the memristor learns. Okay? It means that if I give a sequence of pulses to the memristor, its end state will be different from another sequence of pulses. You have in the brain one structure that does a similar thing. It's called the synapse, right? And we can modulate in memristors short-term plasticity, long-term plasticity, Hebbian learning, a bunch of different paradigms for plasticity using these memristors. So then, why can't we just link neurons and memristors together? So the idea would be to have populations of neurons communicating not through natural synapses, but through memristors. This would give us a head start to start idealizing these medical devices that rely on this analog computing instead of the digital computing. We use our micro-electrode arrays. I don't have, sorry. Same problem as our speakers before. So we use the micro-electrode arrays in a condition where we have different chambers. We place 6 populations of neurons that don't have any physical contact between them. So they are in isolated chambers, and we are going to establish communication between them through the memristors, okay? And we do this in real time. When I say real time, is we have setups that record the activity on the neurons, pass this information to the memristor, and the activity of the memristor is converted to a stimulation that is provided to the other populations. So our goal in this experiment was to do what we can call an activity-dependent bridge. You saw in Anna Sebastian's talk, she talked about network bursts. And what we've done was we wanted to detect automatically network bursts and pass them to the other population. So we don't want to pass isolated spikes. Only when one of the specific populations is emitting network bursts, we want to broadcast these network bursts to the others. I don't have time to explain in detail how we parameterize these memristors, but we can train them as if synapses would be being trained. And the configuration is, I will have a chamber, let's call it the B chamber, and the B chamber is going to be connected to other chambers through this memristor. The network bursts that I was mentioning, and you saw this in Anna's presentation. So this is a network burst, another network burst, and my goal is for the memristor to be able to automatically detect that in an analog computing. So it's an analog device, and it was trained to detect when it was the network burst. So isolated spikes don't trigger outputs on the memristor. Only when there's consistent firing on the population B, then you have this activity. So I'm going to zoom this. So isolated spikes don't trigger these red signals. The red signals are the pulses coming from the memristor. So the synapse made by the memristor is being activated. So only when you have a sequence of consistent spikes signaling a network burst, then you can have the memristor changing state. It goes from a low conductance state to a high conductance state, which means that it now can stimulate, feed forward the activation for the next populations. So, and you can see in here, in blue, you have the population B, and the other lines are activity on the other chambers. And you can see that they become synchronized. We can see this in a different way. So in neuroscience, when you're studying network activity, you tend to use raster plots. Raster plots are, you put on the y-axis the IDs of the neurons, and in the x-axis you put time. So in here you see the activations of several units on the B chamber and several units for C, D, E, and F, so on, so on. And without the memristor, when we have these synapses, the artificial synapses turned off, they are very, very independent, okay? So you see that each network has their own dynamics, generating bursts from time to time, but independently of B. We can see this as raster plot or like frequency, instantaneous frequency. When you see big blue, sharp blue, it means that there's a high frequency of spikes there. When we turn on the memristor and make the memristor available to these networks, then we synchronize all of them. So instead of being 6 networks, they become one network in terms of the network, in terms of network burst activity. So for these conclusions on this type of analysis that we do with these hybrid systems, we are very convinced that these neuromorphic devices can be explored to design new implantable devices that work with analog computing and neuromorphic devices that are very similar in terms of dynamics with neurons. Memristors in particular are very interesting. I just tipped the point of the iceberg. We are doing lots of things with these memristors, and they are very exciting to work with. And we can imagine these systems working on a paradigm where you can have a population that is being monitored, the memristors in an array computing for certain things, and delivering stimulation on a monitor compute actuate paradigm. So you always do these things in teams, of course, and this is my team. I'm very privileged with the team that I have, and in fact, most of them are biomedical engineers, okay? So you choose a very good course for, among all the possibilities, you made a very good decision, because nowadays, most of the fun is at the interface of things. So it's not in the classical disciplines. On the interface is where the innovation is happening. So for the memristors, Domingos, Miguel, Catarina, and the funding. And thanks for your attention.
Okay, thank you so much, Paulo. That was super exciting to see what you're doing with neurons on grids and connecting them with memristors. So let's see what questions we have here for everyone. See if I can go through it. Is it just the 2?
You can ask.
Yeah, no, of course, but the ones that are here are just these ones. Okay. So Mariana has 2 questions, and this is very, I know people are doing this new system, but I think it's nice if people can ask their own questions. Who is Mariana? Do you want to ask your question? Can you, please, go ahead.
Oh, no, you can read it.
Okay. Okay. Last week was discovered the first brain map of an insect. I didn't know that. Well, probably you know. What impact does this have in future research? Does anyone want to address the question? Any one of you? Paulo, you said you do all about
paper
it.
in Science, and yeah, it has 1 week old, and it was in Drosophila.
Right.
I think they eat the larvae, so we are talking about 5,000 years, I think.
So what does the brain map mean? Maybe
It means
that's
that they've done sections of the whole brain, and with electron microscopy, you went to the level of the synapses. So they know
So
for
they
those
describe all the
5,000 neurons, how they are connected. And usually this is very stereotyped. So for C. elegans, it's also, people have mapped also these things. I think it's a very important step, because what the brain does not only depend on the complexity of the units, but also on how the units are connected. So knowing the circuits will help a lot. However, there's very distinct things between, there's the physical circuits, and there's a bunch of neuromodulators that work on top of these circuits. So even if you know the full connectivity,
yeah.
if you turn on a specific neuromodulator, you will highlight a specific component of these highways. If you turn another one, you will have another highway. So it's a fundamental step, but don't be thrilled that everything is solved. There's work for you guys. So, there will be more to be done. Yeah.
Actually, I can't help thinking of these, probably you've all heard of this human brain project that many years ago was started with some similar aim for the human brain. So it seems like an impossible mission, and I think probably it is, as far as we can see. So I think maybe so far they have done something similar to the insect brain for like one column of the cortex, right?
Yeah,
Something like
for
this.
the mouse for one cortical column.
Yeah, but I think the neuroscience community, please tell me if you would agree that this is your impression, would not actually agree that this is the way to go. This is where most of the investments should be, not for the human brain at least. So I don't know if you'd like to comment on this, maybe.
Yes,
Filipa?
absolutely, because even, for example, in the case of pathologies and diseases, right, for sure that we will start mapping control healthy brains. So yeah, of course, the neuromodulators play a big role, and it's very important to also know how the circuits and the pathways are activated and happening. Yeah.
So given that it's very difficult to actually map out everything, so maybe you would agree then that it's not maybe the way to go to learn more about how the brain works.
And it's not the way to go, it's a way to go. Okay, is a way to go.
One way, yeah.
Two way.
Yeah.
So that's the point. I think in science, and there are students that there is also, if you just stick to one way, we will block in some point. If we try to, of course, working in teams, engineers, biochemistries, and all these things. So there are multiple ways to go, and the brain is for sure, the complexity of the brain requires a complex way of going.
Yeah.
But every question has to be very specific. And that's where, so the system is complex, but I think that today we have an example that every question has to be very specific. And the other thing is that really there are surprises in the way that we need to solve one by one. But as a whole, the system is complex and has to be approached by multiple ways. And the error of the human brain is really to think that that was the solution.
Yeah. No, I guess it's just a question of maybe investment. There's limited resources. That's what I meant in the way to go in that sense. Like if we decide, yeah, exactly, in that way. Because of course, I think we as scientists like to have the freedom to ask the questions. So maybe actually connected to this question, it was actually very interesting that Paulo started by saying in neuroscience, the first question is, what's the scale, right? Temporal and spatial. And I had the feeling, with my own bias, obviously, that all of you are actually working at similar scales. Well, maybe Filipa with the whole, right, like very microscopic. Maybe you tell me, right, like very small spatial scale and also very high resolution, at least Paulo and Anna. So would you like to say more about which scale you are working on?
In my particular case, the bulk of what we do is in
vitro. Yeah.
So we work with these microelectrode arrays and microfluidics, where we go not to the level of one single neuron, but we can go with a pipette and say, I will put neurons in here, and I really like to play Lego when I was a kid. So you do this kind of stuff. We also have these projects going on. For example, the Santa Casa Neuroscience project and La Caixa project are at the systems level. And we do this in collaboration with clinicians. But we don't play in the middle. And the thing is that, all these equations that you have been learning for the first years of your undergraduate studies, they break down when you try to go one or two more orders of magnitude. So if you have a very good model for a couple of, let's say, a couple of neurons, it will be very hard for you to use this model to extrapolate behavior like what Filipa has.
Yeah.
So most of our mathematical modeling and computational modeling breaks down when we have more than 1-2 orders of magnitude.
Yeah.
So you need to stick.
Yeah, it's the challenge, right? Maybe we can hear
that's
from.
a big challenge.
I think Anna definitely works in a very.
And the middle is the problem.
The mesoscale. But I like to think that we can work there.
Well, we can approach it, but anyway, we still feel that that's really the middle. Of course, we can understand how a population of neurons work. Even now, with optogenetics or chemogenetics, that is approaching the middle. Really, we can stimulate certain population and see what happens in behavior. So this is really approaching the middle, but that's still lots of things that are still missing between even a circuit as the striatal, the circuit of control of movement, and I think that is quite good knowledge of that particular circuit, and then the behavior that it produces. So really, I'd say that the future is in the middle. But of course, you need to understand both sides. And behavior is similar. So it's also we do nowadays lots of behavior in the lab, but it's stimulating because we see that certain drugs or certain manipulation of certain disease affect specific behavioral patterns. But then it's a black box. Inside, you don't know what is going on. But anyway, this is the challenge in science. So.
Well, maybe Filipa can add something about her own scale of her.
My view is that more than what the scale is more than what the collaboration is. So Paulo already hinted to that at the beginning of the presentation. I think, for example, like neuroscience is not a field where you can only be a biologist or only be a bioengineer. You have to collaborate with everyone, because in order to make meaningful and impactful discoveries, you have to have the whole concept in order to understand the micro, the nano, and also the systems scale. You have to have all. It's really transversal, and it's very important to collaborate with people from all fields. It's very, very multidisciplinary. It's really, and I think that's very interesting because as scientists, we also like to know a bit about other fields. We're not stuck in ours, right? We need to remove the barriers and just be curious about everything that's around us.
Yeah, I'd agree. And actually that links very well with the second question from Mariana, which is about whether you, Filipa, found it difficult to understand the physics and the mechanical aspects of neuroscience with your background in biochemistry.
Well, not really, because in my course at Porto University, we were officially part of the science Faculdade de Ciências, and so we had the physics and mathematics with the people that were like the same. We had this very common disciplines. And then something that was more bio with the people that were studying medicine at ICPES. So I was really exposed to the hardcore things from all perspectives, and I think that really opened my eyes. And so I have a solid background, I will say. Maybe I don't remember a lot of it anymore because, more than 10 years have gone by, but yeah, I feel like it wasn't too hard.
Okay, well, I think most students here, can you maybe tell, who is biological engineering student? Most of them. So who's not? Okay, are you biomedical engineering student? Anyone? No, but some other masters. But yeah, I think because we know the curriculum that we teach here, and I think the biomedical engineering students maybe have more physics and programming, electrical engineering background, which is more different from Filipe's and Anna's and closer to Paulo's and my own. And I think there's a lot of people with our background working in neuroscience, maybe at different levels as well. And I would say that it was never felt as a difficulty to go in this direction, right? So if you have all the math and the physics and the engineering, I would say you can learn a little bit about biology. Filipe is saying that coming a little bit the other way is also not impossible. So I would say that, yes, it's you can.
You mentioned one thing that perhaps that's important, a message for the students. Perhaps she doesn't remember
the biochemistry.
But anyway, that was the seed of other knowledge. And so when we asked students, what's the math? Why am I learning this? Why do I need to learn this? But anyway, that are seeds and layers where the further thinking can be constructed on. And this is very important. So that's something that is multidisciplinary, or I'd say underwater. So
broadband. I think that's the thing.
Broadband studies and type of studies is really important for being creative later on. So
I was absolutely going to mention that because I think what you get out of university, it's not or remember the equations, it's the set of skills and tools to learn how to think. Okay, if you still think that in 30 years you'll remember this equation that you learned this week, this is not going to happen. Only if you remember it each week, because that's how memory works, right? So in neuroscience we know this. But even if that doesn't stick, you remember how to think, how to approach a problem. So you can grab a book, study, and in 2 days you know a bit more about something that's key and fundamental to your research. And that's, I think, the great aspect of having a university course. It's having a set of tools.
I think that also links with Paulo and the fact that we need to think as humans because we have ChatGPT anyway. We can just look up all the course materials. We don't need to know them, but we need to be able to think. And believe me that I think going through all the courses you are going through will help with learning to think.
Something here.
Okay.
Very quickly. So I think this question was also going in the direction of which tools would be relevant for me if I want to pursue a scientific career. Naturally, a good analytical reasoning is fundamental because it allows you to think, even if you don't know the equations, it allows you to think clearly. This implies this, it does not fall into that compartment, and so on. But there's one suggestion that usually I make to my students, which is learn one programming language. That's a biased one. That's very biased. Learn one programming language. Because if you're going to do research and if you want to innovate, you don't want to be pressing the same buttons as other people. You want to make your own buttons. And when I say buttons, I mean your software, your analysis, your analytical tools. You want to be able to talk with your data, ask things that no one else is asking, like I said before. And for that, you need a programming language. If you prefer Python, go for Python or MATLAB or whatever, but you should be skilled at talking with the data. And I think that's becoming more and more fundamental in research. Independently of what you're doing, I would make the same recommendation for social sciences.
Yeah, I think again it comes back to reasoning and a type of reasoning which is oriented to programming a script. Again, ChatGPT will write the code for you. So at this moment, you just need to know what you should ask ChatGPT, and that's why you need to learn one language. Yeah, I agree with you. I'm just saying that eventually no one will be coding, but we need to know what we want the code to do for us. So and that the only way to do it, to know, is to have done it before. I agree with you fully. Yes. I don't know if there's any other last questions from the audience. I think we should be closing the session. So thank you again very much, everyone.
Just before closing the session, I just wanted to say that the winner of the giveaway is Caterina Pina. I don't know if she's here, so you can grab your prize from here. And to finish, I just would like to thank you all for being here. Thank our moderator and our speakers for this very interesting talk and presentations. Thank you.
Thank you.